Hello, and thank you for joining me today. In this presentation, I'll be discussing how oncology biomarker discovery has evolved from what we previously saw as a relatively simple single biomarker approach into what we have today as more complex multi-omic strategies, and of course, why that evolution has been necessary. And I think one of the key elements is that the problem is no longer around generating data. We know we can seek genomes, we can measure proteins, we can look at those circulating immune populations and characterize them. But the challenge is really in determining what data will provide more biological understanding and ultimately support our clinical decision making. So my goal here today is not to suggest that every study needs to be a multi-omic study, but rather when we can look at the question and the problem and determine, is genomics sufficient, or when do we need to add those additional biological layers to inform those clinically meaningful insights? So I'll briefly outline the discussion. First, we'll kind of take a historical look at the evolution of oncology biomarker discovery. And as we'll see with each technology, we gained information, but then what we saw is that we also created an important clinical problem or saw an additional biological limitation that came with each new insight. We'll discuss why multi-omics approaches can provide a more complete picture, especially a more mechanistically informative view of tumor biology, particularly as we started to see the emergence of immunotherapies, ADCs, and cell therapies. And finally, and perhaps most interestingly, we'll look at some practical considerations for designing those robust biomarker workflows, because we need to make sure that not only do we need that biological hypothesis, but we need to be closely examining our specimen selection, pre-analytics, analytical validation, and data integration, and make sure that all of those components are adequately addressed. So to begin, let's take a look at the history of oncology biomarker development. And it's really an impressive story of progress as we look through these successive layers of biological resolution. We can move in this graphic from the left to the right, starting out with morphology. We were starting to just get basic information, ability to identify and classify tumors, but really very limited insight into the molecular biology underlying that tumor itself. We start to move towards more single marker assays, particularly developed through immunohistochemical techniques, and they allowed us to better inform treatment selection. But as I mentioned, they're kind of single-analyte in nature, and cancer biology is rarely driven by one single variable. From there, we moved into genotype-directed therapies. We were able to look at mutations such as EGFR, ALK fusions, and could start to really match that molecular alteration to an appropriate therapeutic intervention. We kind of broadened that as the next step using technologies such as next-gen sequencing, and that allowed us to create more multiplex testing. And I think that was especially critical, as we know, because tumor samples are very limited in nature. And so the importance of gathering as much information as we could from that kind of scarce sample was, a true benefit. Then we moved into liquid biopsy platforms, and kind of one of the critical elements there was the minimally invasive nature of liquid biopsies. And that allowed us to do more kind of steps in detection and that serial molecular monitoring. And then finally, where we're at today, that integrated multi-omic era, where the objective isn't just to catalog mutations, but to really understand that biological state and the mechanism that's driving that disease. So, all important advancements, but as you can see with every generation, we were solving an important problem, but we were also revealing a new blind spot. And so this sequence of limitations is what ultimately drives that need for a more integrated biomarker strategy. And of course, the driver for this need is certainly can be pointed to in the therapies themselves. As therapies have become more sophisticated, of course, the biological and biomarker requirements have evolved accordingly. So again, kind of moving through the therapy development and advancements, we can start with chemotherapy, where it's really a very broad patient selection strategy, and that's appropriate using that kind of treatment. As we develop more targeted therapies, we started to introduce that need for more molecular matching. So even when the correct driver alteration is identified, we were still seeing those resistant mechanisms eventually emerge. As I mentioned previously, immunotherapy, definitely a driver for further complexity. And we have numerous proposed biomarkers there, including PD-L1, looking at tumor mutational burden, perhaps also like digging in on those immune gene signatures, but none of them truly or fully capture that complete immune state. We continue to advance, now moving towards antibody drug conjugates or ADCs and bispecific antibodies. And again, these introduce additional challenges. The target expression frequently exists on a continuum. So rather than just that simple positive negative variable that we may have seen in the past, much more challenging to kind of understand when is that target being displayed and accessible. And then finally, cell therapy is perhaps one of the most complex examples there. The response here depends not just on the tumor characteristics, but also on that immune status. So what is the host immune fitness? What is the immune persistence, clonal dynamics, all putting that together around the treatment context. So as therapeutic mechanisms have become increasingly multidimensional, those biomarker strategies have had to evolve from just that isolated measurement to a more integrated biological assessment. And of course, you have to be cautious. Programs, if they focus too heavily on just the assay selection, they can miss spending enough time upstream, really digging into what is the biological question I'm trying to ask. And of course, the first question that we feel you should be asking is what clinical decision will this biomarker support? Without that really being clearly defined, you can really run into downstream problems regardless of the success that you see in your analytical performance. Next, of course, comes that specimen strategy and pre-analytics. Just need to be asking questions such as, is this the correct matrix to use? Should we be using tissue or plasma or PBMCs or some combination therein? What are the correct collection tubes and processing workflows and all of the stabilization methods that need to be put into place to make sure it's appropriate for that biomarker and the question that you're asking. Then we can move into analytical validation, evaluating those parameters such as sensitivity, precision, reproducibility, interference, and developing those quality control metrics around that. So only after these foundations are established should we start to focus more on the predictive modeling and the validation. And one common issue that creeps up here is that sometimes statistical models can be developed before really the biological assumptions are adequately tested. So it needs to be developed at the appropriate time to make sure that you're not leading your data to perhaps overfitting or poor generalizability. And finally, you need to make sure that you're asking what clinical action is going to follow from this result. Whether it's positive, negative, or indeterminate, you need to make sure that there's an outcome from these implications. And the reality is that many biomarker programs fail not because the assay itself is poor, but because these earlier design decisions weren't adequately addressed or fully optimized. So here's one of the central themes of the presentation. As we look at DNA is really describing what may happen, but integrated biology helps us to understand what's actually happening. So if you think about a genomic profile, it's going to tell us which alterations are present, and that's certainly valuable, but it doesn't necessarily tell us are genes being expressed, what signaling pathways may be active, whether proteins are functioning, or how that surrounding immune microenvironment is going to potentially influence a treatment response. So certainly need to be aware that there can be a tremendous gap between the molecular potential and that biological reality. And that's where the need for those additional analyte layers becomes highly informative, and it really can be built into multi-omic approaches that help to bridge that gap. We have to be careful not to treat biomarkers as static entities. We hear this quite a bit in the field, and it bears repeating here because treatment can and does fundamentally change the biology over time. And we know that tumors can adapt continuously under various therapeutic pressures. Those responses can evolve, perhaps resistance emerges, or immune interactions could shift. And so it's important that you're looking over that continuum, because really a baseline molecular profile, well, it's a good starting point. It probably becomes less representative as the treatment continues. So this is where the advantages that we see in longitudinal liquid biopsy can add certainly significant value to your biomarker program. So serial sampling will allow us to look at those response dynamics, better understand the persistence of therapeutic effects, perhaps see if there's any emergence of resistance pathways or eventual mechanisms of escape. The lower right figure here supports this clinically, and this is actually data that was part of a 2025 meta-analysis where they were looking at advanced non-small cell lung cancer, and they found that ctDNA decrease or clearance is highly predictive of improved survival outcomes. So specifically, ctDNA reduction was significantly associated with better overall survival and progression-free survival. So this just underscores the relevance of that longitudinal plasma ctDNA assessment and predicting survival for patients, particularly in this case, patients with non-small cell lung cancer. So the key lesson and takeaway here is that biology is dynamic and that our biomarker strategies need to increasingly reflect that reality. In this case, RNA provides a fundamentally different perspective than DNA. While DNA is giving us that genomic potential, RNA instead reflects that active transcription. So with RNA profiling, we're enabled to see and identify treatment response signatures, again, kind of get a better view of those emerging resistance pathways and potentially immune biology that may not be readily apparent from DNA analysis alone. And kind of an example of that is two tumors may share identical genomic alterations, but exhibit very drastically different transcriptional states. That said, RNA can present practical challenges. It's inherently less stable than DNA. We often see input quantities are much lower. And so that leads to results that are highly sensitive on those collection methods, stabilization strategies, processing times, all those pre-analytical variables that can come into play. So it's really important when we're looking at RNA that those pre-analytical conditions are very tightly controlled. So the recommendation here is measure RNA when the question concerns that active biology, but be sure you really have a standardized workflow in place before you go ahead and generate any large data sets. And kind of moving through that progression of information that we're getting, if RNA is revealing that biological process, then as we look at proteins, they're really providing that closest approximation of biological function. And this is really critically important as we think about therapeutically relevant changes that occur downstream of transcription. They may be things such as phosphorylation events, perhaps cytokines are being released or receptors are activated, and that can't always be inferred accurately from just genomic or transcriptomic information alone. So this is where proteomics is another essential layer, really building on a better mechanistic understanding. And the example on the right here shows that protein-defined immune states, specifically mature or regulatory dendritic cell states with certain T cell features, these can actually be used to stratify outcomes in patients treated with immune checkpoint inhibitors. So the key point here is that protein and immune cell state measurements can provide outcome relevant biology that DNA alone would not capture. And of course, just like the other analytes with proteins, there is challenges as well. We see that hemolysis can significantly affect measurements. Sometimes we have dynamic range limitations that can remain substantial. And occasionally we're encountering platform to platform variability that can really complicate interpretation or cross-study comparisons. But when the question at hand is really focused on function, then protein is the right analyte to be looking at. And then as we think about immuno-oncology applications, this is where better understanding of the immune system is probably just as important as understanding the tumor itself. And my background is in immune biology, and so this is one of the areas that I find most interesting, and can look at it across these three dimensions. The first is composition. So if you think about that immune population, who is present? What immune cell subsets are we seeing? What's the cellular balance? Are there any suppressive populations in there? The second is clonal engagement. So who's expanding? You can look at TCR and BCR analysis to provide better insights around this, such as antigen-driven responses. You can look at clonal diversity or blood-tumor relationships. And then that third dimension is really focused on function. So what can those cells actually do? What's their activation status? Is there a cytotoxic potential there, proliferative capacity? These are all critical considerations. So one important consideration here is that a response to immunotherapy depends on much more than just the cell count alone. And hopefully I've kind of led you through that. But the immune system, as we think about it, needs to be present, needs to be engaged, needs to be active. And so we need to better understand, the status as it pertains to that. Are we going to get that durable response that's perhaps necessary? So making sure that we're not just focusing on immune enumeration, but really understanding that immune response more holistically and whether or not it can be maintained. And here we're going to start to dig into multi-omics. So while multi-omics can certainly provide that deeper biological insight, it does introduce substantial complexity. And kind of some of the call outs and maybe risks to be aware of are called out here. First being sample volume certainly is a limiting factor. With each analyte that you're trying to measure, it does compete for that finite biological material that you may have access to for the patient. Certainly, pre-analytics becomes more difficult as you're thinking about different analytics. There's typically very different handling conditions for each. And so how do you build that into your experimental design? Assay platforms, they can vary in performance characteristics or sensitivity profiles, and they can certainly be a source of bias. Then as you think about that data integration, of course, that becomes a lot more complex, and we have very heterogeneous biological measurements that need to come all together to address that question at hand. And then when we're thinking about validation requirements, because there are those multiple streams of data, we need to make sure that they're ultimately supported by a unified interpretation strategy. And then most importantly, clinical utility is not guaranteed just because you're using a multi-omics platform or design. And so hopefully I've conveyed by now that not more analytes do not necessarily produce better biomarkers. The objective shouldn't be focused on maximizing that data generation, but should really be focused on addressing the biological question at hand, making sure that's very well defined and that's going to lead to improved decision making. And as we think about multi-omic studies and why those fail, really one of the key factors is investigators underestimating that impact of pre-analytics. So when we're moving from those single analytes to multiple analytes, the complexity really doesn't increase linearly. It's quite dramatic, almost exponential in complexity and scale. So the challenge here is that every analyte class has its own optimal specimen requirements. So conditions that may preserve ctDNA may not really be ideal for RNA integrity. And I think, one of the things we see, especially as a biospecimen provider, is that researchers may try to ask one specimen to support too many objectives simultaneously. If we think about that single blood draw in the case of a liquid biopsy, are we expecting that to provide ctDNA, RNA, proteins, extracellular vesicles, cytokines, PBMCs, all of that in a single collection strategy? It's really important to understand that every collection strategy involves trade-offs. And so starting with the most important endpoint is really where you should be thinking and designing that specimen strategy around that. So if RNA-based response monitoring is really the primary objective, then optimize your experimental design for RNA. If pharmacodynamic protein measurements are critical, then prioritize proteomic integrity. And as we think about multiple analytes, if they're equally important, then consider collecting in parallel so that each of those collections can be optimized for the specific purpose. And so here, the key principle is to think about the perfectly executed assay cannot rescue a compromised specimen. So given all of these complexities, how should we think about designing that optimized multi-omic biomarker program? So the answer is really to ensure that you have a fit for purpose framework. And one of the biggest mistakes we see across translational research is a focus on technology-driven study design. So a new platform becomes available, and I think sometimes the temptation is to measure everything that's possible because the technology allows us to do it. But you have to be really careful here. Generating data, as I said, is not the same thing as generating insight. So the guiding principle I really want to drill home here is that every analyte should have a clearly defined role before the study begins. Every analyte should have a job, every specimen should have a purpose. So tissue biopsy shouldn't simply be collected because tissue is available. It's really important that the specimen is contributing information that can directly address some type of biological uncertainty that exists. And that's where multi-omic programs are most successful. If they're very well disciplined, well defined, they know exactly why each analyte's being measured and how those measurements will be interpreted, because that's how we can really ensure success before any data is generated. So when is multi-omics actually necessary? And this slide kind of hopefully provides you with somewhat of a guide to think about that, perhaps not all the answers, but a framework to consider. And the answer is really not that multi-omics should not be the default. It's certainly a helpful tool, but its value depends on the biological question that you're trying to answer. In many cases, genomics alone can be very effective. And in those settings, especially where the biology of interest is primarily genomic, adding additional analyte layers isn't necessarily going to change the decision. It's really just increasing the complexity. So again, caution, be cautious as you're thinking about that. But where in other situations, the biology may extend beyond DNA and what it alone can tell us, that's where we may need to consider multi-omic strategy. As we're trying to understand or predict immunotherapy response, we know that the mutation profile is important, but doesn't capture that full picture. So a response may depend on the immune cell composition and, as I was mentioning before, that activation state or inflammatory signaling. And so those questions may require a much broader view, looking at the RNA, the protein, the immune profile, in addition to the genomics data. So if you look at the use case triage table below, hopefully this is a helpful framework for you to consider. If you're for mutation screening, when you look there, DNA alone probably is sufficient. If you're thinking about assessment of an immunotherapy response, multi-omics is probably warranted. As you think of early detection, integrated approaches can certainly provide advantages, but you have to be careful here because they do demand the highest bar for analytical and clinical validation. So are you truly getting the information that you need to, again, address that question or make that next decision? So as you think about when to use multi-omics, it should really be driven by what uncertainty remains after that first measurement. And the future of biomarker discovery is certainly shifting from marker detection towards a better understanding of that biological state. And as I've drilled home many times here, the ultimate support for the decision. And the evolution that we're seeing is really in these three major areas. First, as we hone and develop more AI and machine learning enabled models, that allows us to better combine those molecular, pathological, imaging, and clinical information. Second, composite biomarkers that integrate multiple biological features into a unified score are becoming important tools for us to use. And then third, that dynamic longitudinal biomarker monitoring allows us to view those biological changes over time. So importantly, success will depend on validation, on interpretability, and reproducibility, not merely just an algorithmic sophistication. So there's some key principles to consider as we look to what really creates the strongest biomarker program. And it's not looking to measure everything that's possible. The strongest programs are really focused on matching that biomarker strategy to the therapy, biology, the specimen, and ultimately the decision that needs to be made. So as we go through these principles, the first is really focused in on that therapeutic complexity. Certainly, as oncology therapies have become more precise and biologically sophisticated, our biomarker strategies need to evolve and adapt as well. Second, we need to move beyond genomics. And the key point here is when the question requires it. Certainly, DNA remains foundational, but we'll see instances where that may not be sufficient. The third principle is to use multi-omics rationally. Certainly, multi-omics creates a lot of value when each analyte is being used to answer a specific question, and again, addressing that biological uncertainty. Fourth is to design before you collect, really making sure that you've worked through that process before any data generation occurs. And hopefully this is one of the key takeaways that you bring with you is that specimen strategy, consideration around pre-analytics, quality control, and data integration really need to be considered before any patient samples are ever collected. And finally, think dynamically and think in systems. So the future of biomarker development will increasingly be longitudinal, integrated, and multimodal. And we'll start to move away from just those isolated measurements and towards models that better capture biological change over time. So if there's one message I'll leave you with, it's don't maximize analytes, maximize the interpretable biological signal that can change a decision. At BioIVT, our focus is on enabling these types of translational programs through access to those fit-for-purpose specimens and collection strategies. And we do this through a variety of ways. First, providing access to disease-specific cohorts, also access to patients who may be on a wide diversity of different treatments, ensuring that those robust quality control procedures are in place to ensure reproducible data that you can obtain from those specimens, incorporating longitudinal sampling approaches, and then providing that flexibility in the collection designs and approaches. We know there's not any one-size-fits-all approach, and so we really try to work carefully and closely with our clients to understand what their needs are and, where appropriate, develop those customized collection protocols and programs. Our goal is to help investigators generate data that's not only analytically robust, but also biologically meaningful. And with that, we'll close the discussion for today. I hope you found this helpful and informative, and I'd welcome any follow-up questions if you would like to reach out to me at any time.